All right, let's have a little fun. Little review of where we've been and how it's leading into what we're doing. So glucose as fuel. Glucose as fuel. What do we do with it? Where do we start? Glycolysis. So, and we don't care too much about all the individual steps of glycolysis, but what's the main result of the glycolysis? Why do we do glycolysis? Pyruvate. So get pyruvate. So, and what's the immediate payoff? So, gonna get some NADH, although that's not the immediate payoff. That could be a payoff, but what's the immediate energy payoff of glycolysis? Yeah, you get 2 ATP. And that's so backwards. It's supposed to say ADP to ATP. So just keep that in mind. We get 2 ATP. Notice we use up 2 ATP, we produce 4, so we get a net 2 ATP out of glycolysis. That's the immediate payoff. Now, if you're aerobic, there's going to be a much bigger payoff later, right? But that's the immediate payoff. So the much bigger payoff is what we'll talk about now, though. So, oh no, that is correct. We got 2 ATP being used, but 4 ATP being produced. I did have it correctly up there. My bad. So, but notice here, we've produced 2 NADH right here. So if you recall with those NADH, if you are an anaerobic organism, or if you're like, muscle in the middle of a workout and there's not enough oxygen around, so under anaerobic conditions, that's either going to go to lactate or it's going to turn to ethanol, so in carbon dioxide under alcoholic fermentation. And the whole point of that is just to regenerate your NAD+, so that it can go back and do glycolysis another day. So just keep that in mind. So, but now we got another option. If you're aerobic, that NADH could eventually get shuttled off down the road to the citric acid cycle, or actually past the citric acid cycle, but to the electron transport chain. We'll focus on that a little bit later. So, but assuming we're aerobic, we get our 2 pyruvates, we turn those into 2 acetyl-CoAs, we get some more NADH. So, and then the acetyl-CoA shuttles into the Krebs cycle, the TCA cycle, the citric acid cycle, whatever you want to call it. So, couple things that are still important. This is from the last section of material, but we got to keep this in mind. For every acetyl-CoA, what do you get out of the citric acid cycle? You get 1 ATP. What else do you get? 1 FADH, 2, and 3 NADH. Keep that in mind. That's still going to be important on this exam. I know that's last material, but that's still important now. So again, for every acetyl-CoA that you shuttle into the TCA cycle, 1 ATP, via a GTP, but 1 ATP, 1 FADH2, 3 NADHs. So just keep that in mind. All right, so all these lovely FADH2s and NADHs, what do we now do with those? And that's the focus of this current chapter.
Beta oxidation.
What's that?
Beta oxidation.
Ooh, don't go there. Beta oxidation is for lipids, but what do we do? We are going to reoxidize them. So it is oxidation, but it's not beta oxidation. So in this case, we're going to take the NADH and the FADH2, and we're going to take them down here. And I say we're going to take them down there. They're already really in the area, right? All this happens in the mitochondria. So, but we'll take that NADH, we'll reoxidize it to NAD. We'll take the FADH2, and we'll reoxidize it to FAD. So, and in the process, what actually gets reduced? What's the final thing that gets reduced in the whole process of electron transport? Yeah, we're going to reduce oxygen into water. So if you look at the big picture, NADH, FADH2 are both getting oxidized, so oxygen is getting reduced. Now, this is a big, long, drawn-out process through the electron transport chain, but don't forget the big picture. That is the big picture for electron transport. Now, if you notice, so we just made water, but what's the point here? We're hoping to make some energy, and water's not energy. So how is this eventually going to actually lead to energy production? So, well, we're going to get to ATP synthesis, but there we go. The electron transport chain, its only job is to create this proton gradient. We're going to pump protons into the intermembrane space out of the mitochondrial matrix. So that proton gradient is our fuel, it's what powers ATP synthase. So when the protons flow back down the gradient, that fuels ATP synthase to make ATP. So this is the whole point. We use our NADH, our FADH2, so transfer the electrons eventually over to oxygen. It pumps protons across the membrane during the process. So, and that's going to power ATP synthase. So let's break this up. This is your first diagram on your handout. So to what levels do you need to know this? Every level. So we've looked at it now, just the electron transport chain. It starts with NADH and FADH2, it ends with oxygen. Now let's look a little more specifically. So NADH and FADH2 have two different entry points into your electron transport chain. NADH comes in at what we call complex number 1. So FADH2 comes in at complex number 2. So, but once they make it into the chain, then they go to a common place. So from complex 1, they give their electrons to complex 1 for NADH, to complex 2 for FADH2. Then both complexes will give the electrons over to coenzyme Q. So it's in the membrane, and it's totally soluble in the membrane. It can diffuse through the membrane. So, and it'll diffuse over to complex number 3, and transfer the electrons there. So then complex 3 is going to transfer the electrons to cytochrome C. Now cytochrome C is soluble, but not in the membrane. It's soluble in water. So, and in this case, transfer the electrons to cytochrome C. He'll diffuse over to complex 4, another membrane-bound complex. So pass off the electrons there. And finally, it's complex 4 that will actually give the electrons off to oxygen, creating your water. Question?
So for complex 1, where you have the 4 hydrogens going up, is that 4 different NADHs?
No, this is, so just from one NADH, 4 protons will actually end up getting pumped across the membrane. So we'll go in there in the details too next, actually. So if you look for NADH, what's the order for complexes? Where do you start for NADH? 1, then to where? Then to 3, and then to 4. How do you get from 1 to 3? Through who? Through coenzyme Q. How do you get from 3 to 4? Through cytochrome C. So now we're filling in some of the pieces, and we're going to get more detailed as we go. So if you look also, in complex 1, we pump 4 protons across. At complex 3, we pump 4 protons across. And at complex 4, we pump another 2 protons across the membrane. That is important. You need to know that complex 1, 3, and 4 are where we pump across protons, and it's 4, 4, and 2 that get pumped across. Notice complex 2, that's where FADH2's entry point is. And so for FADH2, we start at complex 2. Then where do we go? Which complex is next? Complex 3. And then which complex is next? Complex 4. How do I get from complex 2 to complex 3? Coenzyme Q. How do I get from complex 3 to complex 4? Cytochrome C. Sweet. If you notice, who has a bigger ATP payoff? NADH or FADH2? NADH. Why? More protons across. Notice complex 2 does not pump any protons across the membrane. So in this case, if you look at NADH going through complex 1, so we have 4, 4 plus 2 more, 10 total protons getting pumped across the membrane. For FADH2, since none get pumped across at complex 2, there's only going to be a total of 6 protons pumped across for every FADH2 molecule that enters the electron transport chain. So with less protons pumped across, that's going to ultimately, with ATP synthase, lead to less ATP. So we'll talk about it later, but 2.5 ATPs per NADH, 1.5 ATPs per FADH2. These numbers are approximate. Now, for the purpose of this class, they're exact. So if you were in somebody else's class, though, like a biology class or an upper, 400 level biochemistry class, or somewhere at some other university, you might actually get slightly different numbers. So, but these are the numbers you need to know for this class. So if you learned other numbers for, taking this in bio or cell bio or something, forget those numbers. These are the important numbers now. 2.5 ATP per NADH, 1.5 ATP per FADH2. Cool, we'll make this a little more detailed now. So we'll start with complex 1 and NADH again. So in an electron transport chain, we're just shuttling electrons down the road. So and even within a single complex, there are many electron carriers, and we're just passing the electrons on. So you want to know who all the carriers are in order. It's really annoying. So there are some tricks to get around it. If you look at complex 1, that's where NADH passes on the electrons. So in passing it on to complex 1, who is the first carrier? So FMN. So it's a flavin complex, but it's just FMN, and you're going to want to know that. So an FMN is going to then shuttle the electrons and pass them off to an iron-sulfur center. So when you see FES, that's an iron-sulfur center. So in this case, with NADH, how many electrons is it actually pumping into complex 1? 2. So NADH is putting in 2 electrons. So a lot of the carriers throughout the process will either carry 2 electrons or only 1 electron at a time. So when you see cytochrome, so we're going to get there shortly, but when you see a cytochrome, what is a cytochrome? kind of like heme. So, and what's at the center of it? So we've got some iron at the center of it. And iron's going from +3 to +2, and the cytochromes carry 1 electron at a time, because it's just Fe3+ versus Fe2+. It's the iron at the center of the cytochrome that actually getting oxidized and reduced. So when you see a cytochrome, you should automatically think, oh yeah, 1 electron at a time. So when you see copper down here as well, way over in complex 4, he's going from +1 to +2 as well, and will therefore transfer 1 electron at a time as well. So when you see coenzyme Q, though, coenzyme Q is 2 electrons at a time. The iron-sulfur centers will again be 1 electron at a time. NADH was 2 electrons. FMN is going to be 2 electrons. But anytime you see iron, it's pretty much 1 electron at a time. So in this case, NADH hands them off to FMN inside complex 1, which hands them off to an iron-sulfur center. And then the iron-sulfur center is going to give them to coenzyme Q. And it gives them 1 at a time, but coenzyme Q is going to take 2 electrons. Now, coenzyme Q can take them 1 at a time, but it's going to be transferring 2 electrons, at least net result, over to complex 3. So coenzyme Q, also called ubiquitin. How much did you guys talk about the Q cycle? Much at all? Cool, I didn't think so. We're not going to talk about it either. But there's this cyclic process that happens here. It's just called the Q cycle. But it involves cytochrome B. So it's kind of a separate process off the main chain that's kind of cyclic. So it involves cytochrome B. It is part of complex 3, and that's why there's a cytochrome B. It's actually 2 different cytochrome Bs in there. But if you didn't talk about it, great. But net result, regardless of whether we talk about the Q cycle or not, coenzyme Q is handing off the electrons to complex 3. Initially handing it off to an iron-sulfur center. That iron-sulfur center will then transfer the electrons to cytochrome C1. So if you notice, there's a lot of different cytochromes in here, and we just really try to make it as confusing as possible and name them cytochrome this, cytochrome that, cytochrome this, cytochrome that. But you got to know which is which. It's really important. So you want to know every carrier along the way. So cytochrome C1 will then pass the electrons to the soluble carrier cytochrome C. And that's our link again between complex 3 and complex 4. Cytochrome C hands them off to cytochrome A within complex 4, which then shuttles them off to copper, then to cytochrome A3. And it's a really nice picture of how it's in line, a little more complex than that. But finally then, cytochrome A3 will hand them off to oxygen forming the water. Okay, so somebody just brought up the names, and we will get to those shortly, but those are important. You're going to want to know those names. But you're also going to want to more significantly know why I want you to know those names, as we'll see in a minute. If you look at complex 2, we said complex 2 is where FADH2 feeds into the electron transport chain. So when I say it feeds in, though, it's kind of cheating a little bit. Because FADH2 is actually part of complex 2. It's actually succinate that is feeding into complex 2. Now, where's the succinate from? Yeah, from the citric acid cycle, the TCA cycle. So when succinate is turned into fumarate, in the process, that's where the FADH2 is produced. So the FADH2 that's produced as part of the citric acid cycle is actually produced in complex 2. So it's not like we have to then shuttle it off to complex 2. We had to take our NADH and take NADH, and it has to go over to complex 1. But the FADH2 is already in complex 2. So the FADH then passes on its electrons to an iron-sulfur center, and then again passes them off to coenzyme Q and on down through the rest of the complex to 3 to 4. Same as the rest of the pathway as we did with NADH. Look even more fun? Notice how the diagrams keep getting a little more complex. So here's just another way of representing it. I think this is more confusing, just so you know. So rather than looking at, okay, NADH gets oxidized at the same time, then FMN gets reduced. Then FMN gets oxidized, causing Fe3+ to get reduced. And notice it takes 2 Fe3+ to get to 2 Fe2+ because it's 2 electrons. So on and so forth. So if you look here, this is just kind of saying the same thing. I'm not showing both the oxidized and reduced versions like on this particular diagram, but this one's much easier to memorize if you're trying to memorize this sequence of events from. So just keep that in mind. But this one's going to show a little more of the big picture. And it just shows how in transferring the electrons, the guy who is gaining the electrons is getting what? Reduced. And the guy who's giving away the electrons is getting oxidized. And so this is really going a little more into the redox side of things. So, but if you'll notice, it's the same exact order of events and stuff like that. Got it isolated by complex 1, 3 and 4, as well as 2, 3, and 4 from succinate. And again, want to focus on this just a little bit more. So we just went and looked at a bunch of the trees, but want to step back out and look at the forest again. This is complex 1. Now the electrons are transferred from, NADH off to FMN and off to the iron sulfur center, and then finally off to coenzyme Q to ship them over to complex 3, right? So, but you're going to want to look at complex 1 now as an isolated event. And I want you to look at it as kind of a black box. And when I say black box, what I mean is I don't want you to really get too caught up with what's actually going on inside of complex 1. We just now want to look at the overall net reaction of complex 1. What's happening over here as a result of complex 1? NADH is getting oxidized. So, and at the other end, what's happening to coenzyme Q? He's getting reduced. That is the net reaction for complex 1. NADH gets oxidized, coenzyme Q gets reduced. Yeah, there's some electron carriers along the way inside of complex 1, but the net reaction is also something you just need to know. Notice that's also where it gets its name. If you guys recall, different enzymes often have different names. Kinases are ATP-dependent, phosphorylators. So phosphorylases phosphorylate things in a non-ATP-dependent fashion oftentimes. Phosphatases dephosphorylate things, so on and so forth. Anybody remember what a dehydrogenase was? Oxidation. That's what it does. And in this case, look at the name of the enzyme. What does it say it is? It's NADH's dehydrogenase. It's NADH's oxidizer, and that's half of what it does. So they're naming these things for a reason. One, that'll make it easier to remember the name, but also keep in mind that'll make it easier to remember what's the net reaction here for each of the complexes.
So do people need to know those different areas inside of your box?
So
FMN gets reduced and then the FMN.
most definitely. And if you notice, it's probably the easiest to memorize that in this kind of a sense of just where the electrons are going. Notice NADH hands them off. That means NADH handing them off is getting oxidized. And at the same time, then FMN would be getting reduced. But then when FMN hands them off to the iron sulfur center, FMN's getting oxidized. And FES, the iron sulfur centers, are getting reduced, and so on and so forth. And so it's kind of the same thing, but it's a little easier to memorize the order of steps if you follow this one. One thing to note, complex 1, both things inside complex 1 start with what letter? F. Same as in complex 2, so don't get those confused, but they all start with the letter F. Also, complex 4, everything starts with what letter? C. So if that helps you memorize them, great, but you do got to know the sequence of events here in every single complex. In addition to knowing every single sequence, though, you should just know the net result of each complex as well. And again, complex 1, oxidation of NADH, reduction of coenzyme Q. Let's look at complex 2. So complex 2. So what's feeding into complex 2? So succinate. Sweet. And succinate is getting oxidized to fumarate. So who eventually, at the end of complex 2, ends up with those electrons? Coenzyme Q. So coenzyme Q is getting reduced. And so in this case, notice they call this succinate dehydrogenase. Succinate's oxidizing enzyme. So it makes sense. Again, they name these things in a logical fashion, as it turns out in this case, which is not always the case in biochemistry. So again, know the net reaction though. Succinate oxidized to fumarate, coenzyme Q getting reduced. Complex 3, this is where coenzyme Q feeds in. So coenzyme Q, which has already received the electrons, is now going to hand them off to complex 3. So coenzyme Q is now getting oxidized, and at the end of complex 3, who's receiving those electrons? Cytochrome C. And cytochrome C is therefore getting reduced. And look at the name. Cytochrome C coenzyme Q oxidoreductase. It actually names everything that's going on in the overall reaction. It talks about the cytochrome C that's getting reduced over here. It also talks about the coenzyme Q that's getting oxidized over here. And it's an oxidoreductase, both oxidation and reduction happening. So this is actually a more complete name for the enzyme than any we've seen at this point, so for the entire complex. So it lays the whole thing out. And finally, complex 4. Cytochrome C now is going to hand the electrons off to complex 4, so cytochrome C is getting oxidized. And at the end, so the oxygen is what gets reduced to water. So again, you want to know that net reaction. Cytochrome C gets oxidized, oxygen gets reduced to water. And notice what they call the enzyme, cytochrome C, I shouldn't say C, just cytochrome oxidase. And indeed, cytochrome C is getting oxidized. So the names are there for a reason. So now you see your electron transport chain. You want to know every step in the chain. You want to know what happens, so who's handing stuff off to complex 1, where are they getting handed within complex 1, what the net reaction for complex 1 is. So, and then you want to know, okay, then hands them off so to coenzyme Q, which takes them to complex 3. What's going on inside complex 3? Complex 3 hands them off to cytochrome C. Cytochrome C hands them off to complex 4. What goes on in complex 4? And then finally, oxygen gets reduced to water. And same thing starting with complex 2. Couple things. Really quickly, I missed one thing at the bottom of the first page. If you look at your inhibitors. So you were presented with a few different inhibitors. One for complex 1, one for complex 3, a few for complex 4, and then an uncoupler. So a couple things you need to know. The ones for complex 1 and 3, I'd memorize it, but they're less important than the other two. For complex 4, so if you recall looking at complex 4, in the process of reducing oxygen to water, it has to bind oxygen. And so if you look, what are some of the complex 4 inhibitors?
Cyanide, carbon monoxide, and azide.
Cyanide, carbon monoxide, and the azide ion. Why are they inhibitors of complex 4?
They look a lot like oxygen.
Because they look a lot like oxygen, and complex 4 might bind them instead of oxygen. So they're competitive inhibitors of complex 4. So just keep that in mind. They're inhibitors because they kind of look like O2. Question? No, okay. So, and then you got the uncoupler. There are a couple different types of uncouplers. This is the only one I could find that you guys were presented with. Uncouplers pretty much kill the proton gradient. That's what they do. That's how they function to make all of this worthless, right? If I can do the electron transport chain, but it doesn't create a proton gradient, I can't make any ATP. So uncouplers, you got 2,4-dinitrophenol. 2,4-dinitrophenol is the acid form. 2,4-dinitrophenoxide is the conjugate base form. So what happens when you flood the cell, or system, whatever you're using, with 2,4-dinitrophenol, so it neutralizes the H+ that you're pumping out of the matrix and into the inner membrane space. And by neutralizing it, you don't have a proton gradient. So that's its way of uncoupling. There are other uncouplers that actually form, they make the membrane leaky to protons. And so protons, you pump them across and they leak right back. And they don't leak back through F1, through the ATP synthase. They just leak back across in other places.
I talked to Leffler about the difference between an uncoupler versus a inhibitor. He said uncouplers almost make electron transport more active, whereas the inhibitors almost stop the whole process.
Ooh, that's a great point, actually. So your uncoupler would make electron transport more active, whereas an inhibitor just blocks it. So notice if I bind, instead of oxygen, if I bind some cyanide here, well, if I've bound cyanide, then I can't bind oxygen, and I've shut the whole process down. So however, the whole point of the electron transport chain is what's the immediate goal, again, of electron transport? Proton gradient. And if I can't establish a proton gradient because of an uncoupler, then the cell's going to be like, well, the proton gradient still needs help. Make electron transport go faster and faster and faster, and you still get no proton gradient because the uncoupler's there. That's a great point. So the uncoupler will actually cause electron transport to kind of go in hyperdrive. So whereas like your complex 4 in particular, inhibitors will just shut it down.
That produces heat then? You talked about one thing in metabolism lectures about thermogenin. Is that an uncoupler? So you would use that to produce heat without...
You totally could. So anything that makes the electron transport chain, notice nothing's perfectly efficient, right? We're going to lose some energy somewhere. How do we often lose energy? Heat. And so if I turn the electron transport chain into hyperdrive, like with thermogenin or something like that, but I'm not getting a proton gradient.
You're making heat without energy.
Exactly. I'm making heat without getting any ATP out of it. Cool. Other questions about the inhibitors? Okay. There was one other thing I wanted to mention on page 2. One of the things I cut out that we're not going to cover is oxidative stress and antioxidants. That would include also cytochrome, it would also include P450. It's not because it's not important, it's just something I cut out. So I was limited on time and what I could present, so that's what I cut out. So go back and look over. There's just a couple of slides on that stuff. It's not the most extensive thing, but it was one thing I cut out.
P450 just makes things like cholesterol or things that are hydrophobic more hydrophilic.
So P450 can carry out some funky oxidations and stuff like that. So acting as an antioxidant. So, but it turns out P450 does a boatload of things. Like I read a paper recently about P450 and I was like, wow, this thing catalyzes like 1 million different oxidations and stuff. But technically, you know, it's an enzyme that acts as an antioxidant in some way, shape, or form.